We are talking about RNA transcription. Remember, we've been talking about molecular biology, the central dogma of molecular biology. And just to mention again what we have already said about the central dogma of molecular biology, we say that the central dogma of molecular biology, one, there is you are going to have DNA. So you can convert DNA into another DNA. In other words, you are trying to make a copy of DNA. And then the DNA that you form, you can now convert it to RNA. Okay? To RNA. The RNA that you form, you can now convert it into proteins. So this, my friends, is called the central dogma of molecular biology. So the first step that you can see here, when you get DNA, you make a copy of it. This is known as replication, right? You are trying to replicate, you are making a copy. So that is DNA replication. When you get DNA, you convert into RNA. This is known as transcription. And when you get RNA, you convert it into proteins. This is known as translation. So in the last class, we talked about replication. We are today talking about transcription. In the next class, we are going to be talking about translation. Then we are done with molecular biology. Then other topic we'll do is genetics. Then we are done with the entire biology syllabus. Okay. So now let's start. Really, we've already defined what transcription is. Transcription is basically making RNA from DNA. You see how simple it is? The process of copying a segment of DNA into RNA. Okay, basically, that is what we call RNA transcription. Okay, so what you need to understand is why do we convert DNA to RNA anyways? At the end, we want to make proteins. But then you don't want to make you don't want to use DNA to make proteins. Why? Because any mutation that can happen to DNA is going to cause physical defects. Some of you might have been taking history in high school, and you talked about Second World War, and you did talk about the bomb which was dropped in Nagasaki and Hiroshima. If you remember, those who took history, those bombs which were dropped many years ago, even up to now, they actually still have impact. So you see that someone is just born, they don't have a mouth, or they don't have a nose, or they don't have this. That is because DNA has been affected, and those radiations which are produced by that DNA up to now is actually going to be passed on. All right, so anyone who is born, like say a pregnant woman, someone is pregnant, they are going to be exposed to those same radiations, and the radiations are going to cause damage to a DNA, and damage to DNA is going to form dysfunctional proteins because the DNA is missing some segments. So that is why you don't want to use DNA to form proteins direct. Instead, you want first to convert DNA into RNA. Then after you have RNA, you proteins, because now even when there is a change in RNA segment, it will not be that bad. But if there is a change in DNA, that is very bad. Okay? So DNA, therefore, its function is to store genetic information. It stores genetic information. Okay? Please take note of this. They are very simple, right? And because I'm teaching you without a book, tells you that you can also know these things. So store DNA. Sorry, DNA is going to store genetic material. And then how about RNA? So RNA, as you can see, is the one that we are using to make proteins. So we say RNA transmits. So it's involved in transmission. Okay, transmits genetic material. In case you see those exam questions, then you have already answered right. So DNA is basically for storage, RNA is for transmission. We are transmitting the genetic material which was stored in DNA to make proteins. Say, for example, if you cut your hair, how come you make new ones? That is because you are making proteins. Okay? You are making proteins. Okay, now that we have understood this, remember in this process, transcription, we are talking about the formation of RNA. But then you need to understand that there are different types of RNA. Okay? RNA. And when we talked about nucleic acids, it's there on the drive. In case you are wondering, you can get to follow the recorded session on the drive. So RNA, you have got three types. You have got mRNA, and then you have got rRNA, and then you have got tRNA. You need to understand the function of each one of these guys. So what are the functions of these guys? Now, mRNA carries what we call the genetic code. I love this topic. You are going to understand it very well in translation, genetic code. So what does mRNA do? It carries the genetic code. It is going to be an interesting topic. You don't want to miss out that one. Unfortunately, it's not going to be free like this one. So it is going to carry the genetic code. They'll actually give you the genetic code in the exam, and then they ask you to say what are the proteins which are going to be formed, so you understand anyway. Now rRNA is a component of a ribosome, and you know that ribosomes are used to make proteins, isn't it? And then tRNA, what will be happening is that mRNA is like this is mRNA. M means messenger, okay? And then RNA, the R means ribosomal. And then the T means transfer, so transfer RNA. So what is going to be happening is this is mRNA. What will happen is that rRNA is going to come on mRNA, and then it will start reading. So I believe you guys are in campus. So if you are not seeing, just log out and log in. Your network might be a bit bad, especially if you are at ruins. Okay? If you are not seeing, just log out and log in. So mRNA carries a genetic code. The genetic code is an instruction that is going to tell you what kind of amino acid you're supposed to make. So this is mRNA, and then what I've shaded in blue here is rRNA. So what rRNA will be doing with the ribosome is that it will be moving going forward. As it is moving, it is reading the genetic code found on mRNA. So it is going to be reading nucleotides. Remember we talked of nucleotides, the GCAT, okay, the U, those nucleotides. Except you know that in RNA you don't have thymine, you just have uracil. So let's say, for example, if rRNA reads AUG, this is going to mean A. So what rRNA is going to do is that it's going to tell tRNA to say, tRNA, can you bring an amino acid? So every time you see the base says AUG, it correlates to the amino acid methionine. But this is a topic for this is translation that I'm talking about. We'll talk about translation in the next class, so you don't need to worry much about this. I'm just explaining the types of the RNA and what they do. So what will happen is that tRNA looks like this. They can actually ask you about the structure of tRNA, and then they're going to tell you what is found where. Okay, so this is tRNA. So when rRNA reads AUG on mRNA, it is going to be a signal for tRNA to bring the amino acid methionine. And so on the 3' end, this is the 3' end of tRNA, this is the 5' end. On the 3' end, tRNA is going to carry an amino acid. When it carries this amino acid, it's going to bring it on mRNA, and then it will be making a long chain of amino acids. So a chain of amino acids, a chain of amino acids, and you know that a very long chain of amino acid is called a protein. So that is how we form proteins. But remember that is going to be translation in the next class. Okay? So just take note of the basic functions of the types of RNA. mRNA carries the genetic code. rRNA is part of the ribosome. tRNA carries amino acids. Is there a question? Are we good? Guys, I hate it if people don't respond. Okay. Okay. So now you need to know where the RNA is made. Remember, DNA is found in the nucleus in eukaryotic cells. So therefore, the process of formation of RNA is going to be happening in the nucleus. But the nucleus has got a much darker part. Anyone who knows this part, the darker part of the nucleus, what do you call it? Or the central part of the nucleus? We are telling you to register. These things we've already taught. So you don't want, huh? You'll be surprised. Okay, so the darker part or the middle part here is known as the nucleolus. So when it comes to the formation of RNA, to be specific, rRNA is made in the nucleolus. Please don't forget that. Okay, the nucleolus, of course, is in the nucleus. rRNA is made in the nucleolus. I don't want you to forget that. Okay. So now let's describe the process step by step. As in the last class when we are discussing replication, I mentioned to say all the steps here, replication, transcription, and translation, always start with initiation. After initiation, you are going to go to elongation. Elongation is also known as polymerization, like you are forming a long chain. That is known as the polymerization. And then after you formed a long chain, then you stop. That is known as termination. So that is how we discuss replication, transcription, and translation in these three steps. Okay. So now we are on transcription. Let's start with initiation. How does transcription start? Initiation. Now remember, we are making RNA from DNA. And you guys know that DNA is a double-stranded molecule. All right? What do we mean by that? It's going to have one strand like this. I'm making it in a simple way. You know that the strands are coiled. But don't worry about that, because the enzyme that is going to be uncoiling this thing is going to be doing everything. So one strand runs from the 3' to the 5' direction. Another strand runs from the 5' direction to the 3' direction. Okay. So what should you understand about this? Okay. What should you understand about this? Okay, just give me a second, just 1 second. All right. So I'm saying that for the DNA that you have, you already know that one strand is from the 3' direction to the 5' direction, while another strand is from the 5' direction to the 3' direction. Please take note. Take note that RNA transcription is happening in the nucleus. But then prokaryotic cells don't have a nucleus. So for prokaryotic cells, it's going to be happening where? In the cytoplasm, right? Or the cytosol. Just take note of that. So what happens in the initiation of this process? In initiation, what is going to be happening is that just like in replication, we are the enzyme we are calling DNA polymerase. But in transcription, because we are forming RNA, we have got an enzyme which we call RNA polymerase. Okay, RNA polymerase, different from DNA. In replication, we said that the enzyme that is going to be separating these bonds between the one chain and the other chain is helicase. But that is different from this process. RNA polymerase is going to be separating the strand and also making a new strand. It is doing all the work together alone. Okay, please take note of that. That's very important. Okay. So what is going to be happening is that there's going to be a group of proteins which is known as the initiation factor that is going to move along DNA. So here is a group, let's say, let me just put a group here. This is a group of proteins. And what's the name of these guys? These are known as the initiation factor. I'll just put IF, initiation factor. So these guys are moving on the strand of DNA. Now they are going to be moving going forward like that. Not until they reaches what is known as the promoter. Okay, the promoter region. So what is the importance of the promoter? The promoter is a in replication, we talked about the origin of replication. That is where helicases started. Actually, that's where DNA started separating the strand. But in transcription, the separation of the strand is happening where? On the promoter. I don't want you to forget that, all right? It's happening on the promoter. Okay. Now, what are we doing on the promoter? What is found on the promoter? So a promoter is just a sequence. Okay, it's a sequence of nucleotide, and it is rich in thymine and adenine. You are going to find what we call the Tata box there. Tata box. Why are we even calling it that? So it's not, guys, it's not something to master. Let me make it simple for you. Why is it that this process, why is it that initiation is happening on the promoter? Because you know that between thymine and adenine, you have got two hydrogen bonds. Between guanine and cytosine, you have got three hydrogen bonds. So, Joyce, which one is easier to separate, two hydrogen bonds or three hydrogen bonds? Joyce doesn't want to answer. Anyone who can answer, which one is easier to separate? So two bonds are easier to separate. That is why the promoter is rich in thymine and adenine. That's why we are going to find what we call as the Tata box. Okay, the Tata box. This is just represent telling you the amino acids found there. Okay, not the amino acid, but the nucleotides found there. So the promoter is rich in thymine and adenine. Okay, I've taken time to explain that. So what is happening is that a group of proteins, the initiation factor is moving along the DNA strand, and when it locates the promoter region, which is basically a special sequence or on the DNA, what is going to happen is that it's going to signal RNA polymerase to come and join also. And so there's going to be a protein complex being formed. Okay, so the initiation factor and RNA polymerase will then join together. So now let me write that again. So what is going to be happening is that the RNA polymerase is going to be separating the strand, at the same time is going to be making a new strand. So here is our initiation factor and RNA polymerase. So this is RNA polymerase. Okay, there. So this RNA polymerase is going to be moving forward. And what is going to be happening as it moves forward? It is separating the strand. Okay, is separating the strand. And what are you going to be able to see? You will see something like this. You see, the process is not that different from replication that we already did discuss. So this is the chain. And if you miss anything here, please ask. Please ask. You don't want to have questions in the exam, because remember there is negative marking. Imagine you get 50 correct and then you get 50 negatives. It means that you would have gotten a 25%. Okay, so this is a DNA, and then our RNA polymerase has separated it. Here is a guy RNA polymerase. So that is called initiation. You can see how simple initiation is. Initiation is basically separating the DNA. Okay, just separating the DNA. Is there any question? Nothing. Okay, so once RNA polymerase unwinds, yes, Joyce. Okay, so helicase is not involved in transcription. What is going to be separating the strand is RNA polymerase. At the same time, is also making a new strand. Okay, so helicase is only involved in DNA replication. That's okay. Good. All right. Please don't confuse them. Here, the enzyme involved is RNA polymerase. In DNA replication, it is DNA polymerase. Don't confuse them, all right? It will cost you a lot if you do confuse them. So now we have got two strands here. Immediately you separate the strand, you are done with initiation. Immediately you separate the strand, you are done with initiation. So what is going to be happening is that now the RNA polymerase is going to be making a new strand. But then what you need to take note of is that the strand that is running from 3 prime to 5 prime, if it is DNA replication, we are calling that as the leading strand. Okay, but this is transcription. The strand running from the 3 prime to 5 prime, this is known as the, it has got a lot of names. Antisense strand. Apart from antisense, it is also known as the template strand. Apart from template, it is also known as a non-coding strand. Don't forget these three names, please. Whatsoever you do, never forget these three names. It's known as antisense, template, or non-coding. The other strand is just the opposite of the names that we've mentioned. The one running from 5 prime to 3 prime is known as the sense strand or non-template strand. Non-template strand, also known as a coding strand. These are the three names. Never forget that, alright? Never, never forget. So what is going to be happening is that in RNA, in transcription, remember RNA, this guy RNA is single stranded. It's not like DNA is single stranded. So we only use one strand to make a chain of RNA. We don't use both strands like the way we are doing with DNA. Only the antisense or the template. That's why we call it template, because we are using it to make mRNA. It's a template. Don't forget the direction. Okay. So what is going to be happening is that once RNA polymerase unwinds the DNA, it is going to create this bubble that you can see. But then for this bubble, there is no SSB protein. Remember the function of SSB protein? We said that the SSB protein was there to do what? Anyone who remembers? Yes. So very good. That is the function. But then here you're actually forming a bubble because that guy is not there. Okay. So the size we are separating, it is still coming back again. That is the difference you need to take note again. So here is our compound. There. This is 3 prime and then the end is 5 prime. Remember our interest is only on the template, the non-coding, okay, or the antisense. That's our only interest. We are not interested in the other strand. So what is going to be happening is that as the RNA polymerase is going to be moving forward, it is going to be making a new chain. And it only makes a new chain on the same template strand. So it is moving, it is creating a chain like that. It's creating a chain like that. And then it will continue moving. So if let's say on the template, say you have got 3 prime, A, T, G, C, C, G, C, T, A, 5 prime. They are going to ask you in the exam what is going to be the strand of mRNA that is going to be produced. And take note that what you form in this process is mRNA. Okay. And basically, what we are discussing here is prokaryotic. Okay, prokaryotic transcription. By the way, the group of initiation factors in prokaryotic cells, we also have what we call the sigma factor. The sigma factor will be will be involved in initiation, separating the strand. All right. So what is going to happen here is that the new mRNA we're going to form is going to be 5 prime. A will pair up with what? Anyone? A pairs up with what? So A pairs up with you. Remember, you don't have T. You are forming RNA. You don't have T in mRNA. And then T will still pair up with A. G and C, GC, they always pair up together. So G with C, C with G, C with G, G with C, C with G, but T with U. A not with you. A with what? Oh, sorry. T is supposed to pair up with A. Okay. And then A pairs up with U. A also with U. In other words, and then 5 prime, you are going to have 3 prime. In DNA, you need to know that A pairs up with T. Okay. But then in RNA, you don't have T. So therefore, A is going to be pairing up with U. But if you have got in DNA, if you have got T, T will pair up with what? With A. Okay. But you don't have again T in RNA. So if you had the T in DNA, if you had the T in DNA, it will be pairing up with you. If you had not with you with A still. Okay. If you had the T in the DNA, you want to form RNA, it will still pair up with A. But if you add A in DNA, it will not pair up with T. It will not, because you don't have T in RNA. Instead, it will be U. Is that clear? Okay. So elongation. Okay, so this that is going to continue and will be increasing the chain. That is going to be elongation. And when we are done with that, we have to terminate the chain because we are done already with elongation. So we go now to termination. So what happens in termination? Please take note the strand that you are forming. The strand that you form is always from the 5 prime to the 3 prime direction because the strand that you use is always from the 3 prime to 5 prime. Take note of that. So in termination, what is going to happen is that you are going to read the sequence of nucleotides, okay, that are going to say stop. So when RNA polymerase reaches the termination sequence, special proteins may assist in the disassociation. Okay, so we really don't know the sequence that we are going to reach for termination to happen, but then there is a special sequence that you are going to reach that is going to say, okay, it is enough, stop making the chain. So there are actually two methods through which termination can happen. One method by which it can happen is that you can use something known as raw. Okay, so this protein row can be involved in termination. That is one thing. Okay, it's a special protein. So at the end of transcription, you are going to have that protein, and then it is going to stop. It's going to signal, okay, it is enough. And then another method is that you can reach the termination sequence. The termination sequence. Let's say this was your chain you are forming. At the end of it, or remember you are forming, you are producing from 5 prime to 3 prime. At the end of it, or you are going to form what, there's going to be a sequence that is going to code for what we call a hairspin, something like that at the end. So when you just code for that, that is going to stop the process. So that same special sequence that you've read that is going to code for the hairspin is going to stop the process. So these are the two methods of termination. Now after termination has happened, you have formed your mRNA. But now the mRNA that you have formed is a bit interesting. It's like this. Okay, so this is our mRNA, messenger mRNA. And the blue guys, let's call them exons. Okay, and then the other guys, the red ones, let's call them introns. Now the mRNA that you have formed, not all the bases are necessary. So the when you form mRNA, is going to undergo modification after transcription. So this is known as post transcriptional because it's after transcription. So post transcriptional modification. This is very important. Okay, post transcriptional modification. There are basically three important things that happen in post transcriptional modification. One thing is that you are going to be removing the introns and connecting together the exons. So that is known as splicing of exons. So what is splicing of exons? What you are going to see is that the guys that you can see in red are going to be removed and thrown, and then the blue ones are going to be connected. So you now just have blue ones like that. So that is splicing of axons. Please never forget that. Remember that introns are removed and thrown away while axons are connected together, and this is the mRNA that you want. That is the good RNA. And then another modification that happens is that there's going to be what we call 5' guanosine triphosphate cap. Triphosphate cap. Now remember that the guy that we have formed, the mRNA that we have formed has got two ends. It has got the 5' end, and then it has got the 3' end. Okay, like that. So what is going to happen is that to the 5' end of this guy that we have formed, we are going to add the guanosine triphosphate, GTP. Okay, we are going to add guanosine triphosphate. So it says guanosine triphosphate. But immediately you add guanosine triphosphate, the guanosine itself at the 7th position of the guanine, you are going to add the methyl. Okay, so this is also known as so the guanosine, the guanine. Take note of this. The guanosine nucleotide is quickly methylated at the 7th position. So this is also known as 7 methyl guanosine triphosphate cap. 7 methyl guanosine triphosphate cap. I don't want you to forget that. Please never forget. When you add the guanosine at the 5th position of the mRNA, the guanosine nucleotide is quickly on the 7th position. You add a methyl. Okay. And then the third modification that happens, number 3. There are only 3 modifications. The third modification is known as polyadenylation of 3' end. Now the 3' end is also known as the tail. So this is at the 3' end. We are going to add a lot of adenines. All right, we are going to add what we call a polyadenylation, a lot of adenines. So we're going to see a lot of adenines, a lot of them, a lot of them, a lot of them, a lot of them. They are really a lot, like 200 to 250. So these are the 3 modifications that happen. On the tail, there is polyadenylation, and the tail is the 3' end, remember. And then on the head, there is addition of 7 methylguanosine triphosphate, and then there is a splicing of the exons. Is this okay? Are we good? Okay, let's go through exam questions. That's the only way I can tell if people are understanding. Okay. Which of the following features is common to both DNA replication and transcription? Nucleotides are added to the 5' end of the newly synthesized strand. Deoxyribonucleotides are incorporated into the growing sequence. Ribonucleotides are added to the 3' end of the growing strand. Both require a primer. Both are initiated at the promoter site. Which one can you pick? Mhm. Okay, let's start with option 1. Nucleotides are added to the 5' end of the newly synthesized strand. Is that true? Remember when we are forming the strand, we are forming the strand in the 5' to the 3' direction. So we are adding new nucleotides to the 3' end. So this is out. And then number 2, deoxyribonucleotides are incorporated into the growing sequence. Is that true? Maybe people don't even know deoxyribonucleic. We are forming RNA in transcription, so we can't be adding deoxyribonucleotides. These are nucleotides for DNA, like DNA nucleotides. That's deoxyribonucleotides. That can't be true, guys. Okay, we'll be adding ribonucleotides in transcription, and then three ribonucleotides are added to the three prime end of the growing strand. Is that true? And this is also not true, because in DNA you are going to be adding deoxyribonucleotides, not ribonucleotides. Both require a primer. A primer is basically where you are starting, so that is supposed to be true. Both are initiated at the promoter region. Of course, the promoter is going to be initiation for transcription, but for translation, it is at the origin of replication. All right. So hope with that is okay. Let's go. If the sequence of bases on the template strand of DNA is the 3 prime, 3 prime, T, C, C, G, A, T, 5 prime. The sequence of bases on the mRNA transcript will be. Okay, now you'll be telling me what I should write. Down, this is how you answer these questions. Before you even look at the solutions or the answers, first write it yourself. Down the 3 prime, what am I going to put? 5 prime. And then T is going to pair up with what? With A, very good. And then C with what? With G. And then the other C with G. And then G with what? With C. And then A with what? With U, good. And then T with what? With A. And then at the end, what are we going to put? 3 prime. Okay, like that. So, which one do you think is going to be which one is the answer? Isn't it? Determine the number of amino acids that would be specified from the mRNA sequence. Okay, this one, no, this is translation. When you have got three bases, that's one amino acid, three bases, one amino acid, three bases, one amino acid. So this will be three amino acids, but that's the next topic. The DNA strand which is transcribed into RNA is the dash strand, complementary no sense template double sugar phosphate. Ah, very good. I like that. I like it that you are quick. This means that you have data. So this is how simple exams are. If they give you an exam like this one, this is 3 hours. You spend 2 hours. Just know that the data is small, because you finish within an hour. Okay. Let's go. Which of the following statement is correct about RNA polymerase? It carries out replication of DNA. Is that true? No. It is establishes hydrogen bonds between nucleotides in double-stranded DNA. Is that true? No, it's not in DNA. It acts as an initiation factor during transcription. Is that true? Okay, let me put a dot for you there. It synthesizes both introns and exons. Is that true? This is true. Remember, we said it will make both, and then at the end, we'll now just remove the introns. Do you get that? Okay, so that is the answer. For three, it acts as an initiation factor. It doesn't act. It will combine with the initiation factor to help in initiation. Doesn't act. The process of dash cuts out introns from the primary mRNA transcript and joins the exons together. RNA synthesis, RNA translocation, RNA elongation, splicing, initiation. Which one? Splicing. Very good. During which stage of bacterial transcription is the sigma factor involved? Elongation, termination, RNA capping, splicing, initiation. Is what? You guys, you can't tell me you've forgotten. I even wrote it here. I even wrote it here. Initiation. All right? Yes. Okay. Next. Mature mRNA. Now, what we call mature mRNA is after those three modifications. The first thing that we form is known as the immature, all right, or the primary. But the mature is after modification. Mature mRNA from eukaryotics would contain each of the following features except exons, 5' cap, 3' poly A tail, introns, and exons. Which one can you pick? Which one? Five or four? Okay, four. So would contain the following features except. Okay, so it can't contain introns. Okay, that is true. Five is saying introns and exons. Obviously, when you are maybe yet splicing, but you know that after splicing already you can't contain introns, but exons qualify the answer. The presence of methylated guanine head on an RNA molecule indicates exons which must be removed, a newly synthesized tRNA molecule, the initiation of transcription, a mature tRNA molecule, a mature mRNA molecule. Very good. Ribosomal RNA A is synthesized in the prokaryotic nucleolus, combines with tRNA to form ribosome, synthesized in the eukaryotic cytoplasm, synthesized in the eukaryotic nucleolus, carries a genetic code. Which one do you pick here? Combines with tRNA to form ribosomes. Okay. Anyone else? Are we all thinking the same? Ribosomal RNA, guys. I meant. Let me even go back so that you don't say I'm just remembering. Look at this. What did I mention about this? What do you remember? You can't remember anything. Are you able? It's made in the nucleolus. Now remember, prokaryotic cells don't have a nucleus, so they can't have a nucleolus. So it's made in the eukaryotic nucleolus. It is only in the cytoplasm when it is for the prokaryotic cell. Okay, but for eukaryotic cells, it's in the nucleolus. Is that okay? The process of removing the dash from pre-mRNA and joining the remaining dash together to form a mature mRNA molecule is called dash. Which one do you remove? So we will first want what you remove and then what you join, and what is the process called? So which option can you pick? It's not hard. Very good. Remember, you remove introns, you connect exons. That's post-transcriptional. Post-translation or translation is the making of proteins. We are from talking about transcription, remember? Do you get it? Okay. The process involved in the formation of RNA from a DNA template is called transcription, translation, replication, transformation, protein synthesis. Which one? Transcription. Okay. The DNA template for protein synthesis has the following bases: A, G, C, T, T, C, G, A. Determine the order of complementary base in mRNA. What is A going to pair up with? With you. Do you get it? And then G will pair up with what? With C, and then C, and then T, A, and then G, C, U, isn't it? So which one is the answer? Okay, number four. Transcription occurs along a DNA template in the direction dash, forming mRNA in the direction dash. Uh-huh. Which one can you pick? Two. It occurs in the 5 prime to 3 prime, forming 3 prime to 5 prime. Is that true? Three. Good. 3 prime to 5 prime, forming 5 prime to 3 prime. This is the answer. Which of the following is true about RNA synthesis? RNA synthesis is always in the 5 prime to 3 prime direction. Is that true? Is that true? Let me put a dot. RNA synthesis is always in the 3 prime to 5 prime direction. Is that true? This is not true. The correct one is this one, guys. You are making the chain in the 5 prime to 3 prime. The one that you are reading is the one running in the 3 prime to 5 prime. Is that okay? Okay. Which of the following best describes the cap modification of eukaryotic mRNA? A modified guanine nucleotide added to the 3 prime end of the transcript. Is that true? Not to the 3 prime. A modified guanine nucleotide added to the 5 prime end of the transcript. Is that true? Okay, so it is a modified guanine. These others are wrong because it's adenine. And that's not a cap. That's a tail for the adenine. Which of the following statements regarding splicing of the eukaryotic primary RNA transcript is correct? Exons are removed and introns are sustained in the mature mRNA transcript. Is that true? False. Introns are removed and exons are retained in the mature tRNA transcript. Is that true? Okay, because of tRNA. So please be careful. Huh? Introns are removed and exons are retained in the mature. What's this? IRNA. Is that true? Splicing takes place in the cytoplasm. Is that true? Okay. Splicing takes place in the nucleus. Is that true? Okay, so what you need to understand is this: when you make the RNA, which is known as a pre-mRNA, it starts to undergo modification. Before, remember, for eukaryotes, it is still in the nucleus. Okay, so we are converting DNA into RNA in the nucleus because that's where DNA is for prokaryotes, for eukaryotes. And then after then, when you form the RNA, it is going to undergo modification. Okay, it's going to undergo modification and then it can leave the nucleus. So modification is happening where? In the nucleus. This is eukaryotic. If it was prokaryotic, it would be in the cytoplasm. A particular triplet of bases in the template strand of DNA is a 3 prime A G T 5 prime. The corresponding codon for the mRNA transcribed is be telling me what am I going to put down? Uh-huh. What will be the next? A pairs up with U, and then G, C, and then T, and then at the end, 3 prime. So which one is the answer? So A is correct. Processing of eukaryotic primary mRNA transcripts involves the addition of adenine nucleotide on the 5 prime end. Is that true? No. Adenine at the 5 prime. That's not correct. A methylated adenine on the 5 prime end. Is that true? Many guanine nucleotides on the 3 prime end. Is that true? A methylated guanine on the 5 prime end. Is that true? How about many uracil nucleotides on the 3 prime end? Okay. And the last question. Arrange the following events in eukaryotic mRNA synthesis in the correct order. So you have got A being polyadenylation, B being termination, C being splicing, and then D being capping, E being initiation. So which one is going to be the first one? Which letter is going to be the first? Initiation. Okay. Which one is going to be the next? Polyadenylation, termination, splicing. Which one is going to be the next? Or capping. Now you can see that polyadenylation, splicing, and capping, these are post-transcriptional modification. They happen after termination. So the next one should be B. That's when you can go to those guys. And if you don't know the order, just go to the option that has got EB. And then the rest, it's their order like that. So it's EB. This is the answer. You might not necessarily know the order in which the post-transcriptional modifications will happen. Okay, but for here, they've just put that the first one is going to be the capping, and then the adenylation, and then the splicing. But it doesn't matter really. What you wanted to understand is just the first two. Is that okay? All right. So here is the end. Here is the end. In case you don't want to miss out on translation, you can get to register with us. Remember, exams are soon. You can get to inbox me on that line, and then you get registered so that you don't miss out anything. Otherwise, thank you so much. Enjoy your night.